Let me just say good morning and welcome to all of you to the Proteinopathies ZOOMinar series hosted by the University of Michigan. Today we have the pleasure to host as invited speakers Professor Frederic Rousseau from the Switch Laboratory of Catholic University of Leuven, Belgium, and Dr. Wenwei Zheng, Arizona State University. As a short introduction to Professor Rousseau, I would say that Professor Rousseau obtained the PhD in chemistry at the University of Cambridge in 2002. He is now full professor at the Department of Cellular and Molecular Medicine at the University of Leuven, Belgium. In 2018, he received the Jefferies Lifestars Prize for the most innovative European private finance race. And he is inventor of 13 patents, some of which are licensed to some big pharma companies as Sanofi Aventis and Boehringer Ingelheim. The focus of Professor Frederic Rousseau research activity is related to amyloid aggregation, stability, and morphology. Today, we'll give a lecture entitled Heterotypic Amyloid Interactions, Modulate Amyloid Assembly and Structure. Please post your questions in the QA box, and I will read them at the end of the talk. Please, Frederic, the stage is yours.
Thank you very much for your introduction and for the invitation. Obviously, it's a really nice ZOOMinar series, and it's really great to be able to be here. So indeed, what I want to talk about today is some of the more recent work in our lab, which is about looking at how amyloids and amyloid conformations in particular interact with other proteins and how it affects their behavior. But before I start, first would like to thank all the people of the lab who have been participating in the work you will be seeing. Most of the people on there participated to some of the aspects that I will present, and I will highlight specific people on slides to link them directly to the part of their work. I also would like to thank our funders, of course, and without further waiting, I'll start with the talk. And actually, what I will start with, is a slide whom I used to start many talks already 15 years or so ago as a way of introducing aggregation-prone regions. And so the slide generally goes in the following manner. Proteins are folded, and then, during translation, translocation, stress by mutations, etc., they can unfold. And by unfolding, they actually display specific segments in their sequence, which we call APRs. And these APRs are segments that like to interact with identical segments in other molecules to form beta-sheeted, beta-structured aggregates, which can be amyloid or amorphous, but generally are enriched then in this beta structure. And so that's how the general introduction of APRs went. And we and many other labs have made a whole series of predictors, so how to identify in a primary structure of a protein those regions that are particularly prone to engage in such interactions. And we in our lab developed Tango and later Waltz and even more recently Cordax. Tango, for instance, the first one was based on biophysical principles. So what Tango takes into consideration is beta sheet propensity, charge, hydrophobicity. Waltz later was used when more data was available. We used actually sequence information to predict APRs in proteins, and more recently we used structural information in Cordax. And so But the general logic is the same. It's, you know, again, finding those short, and generally short, we mean 5 to 10 amino acids, something like that, segments in proteins that really like to aggregate. And those are aggregation prone regions. Of course, in the time we were first doing this, so beginning of the years 2000, 2002, 2005, etc., and until very recently, most of the structural information was on such APRs that were identified in a whole series of amyloid proteins, disease-related, but also functionals. And here, of course, you know, this famous picture of the David Eisenberg group. And we and others, Louis Serpel as well, solved a lot of the structures of these APRs. And yeah, I mean, again, this was this logic of these short segments that can then assemble in a cross-beta structure and that pack against each other using dry steric zippers, like they are called. Now, of course, since 2017, and actually by solid state NMR earlier, we started having all those very nice amyloid core structures. And we have a lot of those structures now. And yeah, the question arises whether this APR aggregation prone region thinking is still relevant. Because of course, as you see, and as we know, amyloid cores are formed by much more residues than just the predicted APRs. That's the first thing. A second thing is that the way in which these APR pack and form tertiary structures into real amyloid cores is not by homotypic interactions. It's not the same part, the same segment that sticks to each other, but it's actually packing against another region of the protein in the amyloid conformation. So again, yeah, are these APRs still relevant? Do they still mean something, or is it just you know, a concept that is now outdated? Well, for one thing, we know that these APRs are in any case important for the kinetics of amyloid assembly, as for instance, you can just abolish here A-beta aggregation by just targeting each of the two APRs in this peptide by a single point mutant that abrogates the aggregation propensity of this region, for instance, here by phenylalanine to serine mutation, and here a glycine to aspartate mutation in the second APR. And just by those two mutants, you can entirely abrogate the assembly of A-beta. So indeed, those regions still are crucial, at least for the kinetics of assembly. I will come back to that later, but looking a bit more now at all these nice amyloid structures we have, and here you see a number of polymorphs of tau solved by the Scheres and Goddard labs. And okay, what we did is we took Foldex, an empirical force field in which we participated to the development of many years ago with the lab of Luis Serrano. And we use this tool just to see, now that we have those structures, how much each of the residues in these structures participate to the stability of the amyloid. It's a computational analysis, but the force field is quite reliable. And so it really can give you an idea of what's happening there. And the first thing you can see immediately is that, in fact, not the entire amyloid core is homogeneously stable. You have regions that have sort of neutral contribution to the thermodynamic stability of the fibril. You have some that are stabilizing, and then you have others that are really not happy in this conformation, actually. And if you compare you know, the amount of residues that are either stabilizing, neutral, or destabilizing to these structures, and you compare them to what it is in globular proteins, you can here see that the distribution is more or less similar in the two types of structures. So in that sense, amyloids are just another fold. So you have about 30% of residues that really stabilize globular protein cores. And the same is true for amyloid cores. And then you have quite some residues that are neutral, and you have some that are really not happy. So it looks like, even though many more residues than APRs are inside the amyloid core, really these regions are APRs, and the other ones, some of them really seem to be really frustrated in there. And you can say, okay, I don't believe Foldex too much, I just don't want to trust it. Then you can just look at structural parameters that have nothing to do with Foldex. And for instance, here you can see a comparison of the Ramachandran plots of all the structures, amyloid structures we have now together. And you can see, you know, the 5 psi angles of the more stabilizing regions. And you can see that the ones that are neutral or destabilizing have less well-defined dihedral angles. You have much more variability, and they are deviating much more from the let's say, the canonical cross-beta 5 psi angles. You can also see that the stabilizing residues, in general, the side chains have a better burial than the neutral ones and the destabilizing ones. And the same is true for the backbone H bonds. I mean, they are generally better formed in the stabilizing ones than in the neutral or destabilizing ones. And you can really see, if you look at the structures, that the regions that are more stable, the H bonds, have a much better geometry, while the ones that are neutral or destabilizing really have a more strained H bond, backbone H bonding. And so we looked a little bit more then, okay, if there are regions that are more stable and other regions that are less stable, which one are those? And are those then different in different polymorphs? How do they, how do they vary between polymorphs? And so what we did here is we looked at all the structures available for tau. And we just made a thermodynamic profile. So again, this is done with Foldex, and this is showing the lower it is, the more stabilizing these regions are in the different polymorphs. And okay, it's a bit a crowded image here, but what you can see is that, in fact, for many polymorphs, you have more or less the same regions that are stabilizing and more or less the same regions that are destabilizing. And here you can see how many of the residues in the cores are involved in H bonding. And you can see that there are gaps in there, obviously. But then if you would go and look at the energies, you would see that all these H bonds, even though they are there, they are not actually as good over the entire sequence. So again, it looks that there are regions that are stable, others that are more frustrated. But what's interesting is that indeed, across polymorphs, even though those structures look different, you have the same regions that are happy and unhappy. And here you see the same plot, in fact, just by superposing the energies of the different polymorphs. And again, here you can see that some regions really are important in general in most of the polymorphs, and others are, again, destabilized. And so you can say again, okay, but this is Foldex. So, I mean, it's a computational assessment of the thermodynamic stability, what happens in real life. So we did a simple experiment. So we made, for the whole tau amyloid core, we made peptides with a sliding window over the entire sequence. And then those peptides, we incubated them and we just stained them with THT or PFTAA to see which segments can form amyloid structures on their own, outside of the context of the full-length core. And as you can see, most of the regions we find to be structurally stable can in fact form amyloid structure as a peptide, again suggesting that these Foldex predictions are quite accurate. And of course, this region, for those of you who work a lot with tau, is quite known. It's PHF6. This is PHF6 star. But you can see there are other regions as well. And in fact, it's quite interesting. We could, in fact, predict new APR regions that were not known already in tau. And one of those we looked into more detail in collaboration with Sheena Radford. And here you can see some of the structures. And what's interesting is actually that this region, for instance, forms amyloids on its own. But solving the structure of this peptide, we saw that it actually is very polymorphic on its own. And a story we just published on, we just put on bioRxiv, shows that actually the polymorphs that the conformations that the peptide on its own can achieve are very similar to the ones it adopts in the full-length amyloid core. So this is really an APR again. But what's very interesting about this one is that it's, really suggests that it somehow steers the possibility of polymorphisms in the different tau cores that we know. So suggesting that maybe polymorphism is for sure determined to a large extent by the environment in which it grows, but also that there are, of course, intrinsic structural propensities that are proper to the tau sequence and that restrict the polymorphic landscape that it can adopt. No, so these analyses I've been showing you before, and the fact that you have some regions that are really always important in all polymorphs, to us suggests that maybe there's a sort of framework polymorphism, logic to polymorphism, so that APRs still are the important parts driving the assembly. But of course, due to the fact that between APRs, the segments there are relatively short, the formation of these cross-beta interactions sort of coerce other regions into the core, even if they don't like to be so much in cross-beta conformation. And this coercion occurs because, of course, these regions here then pack against the less favorable regions, sort of stabilizing them. And then you can do this in alternative forms because, of course, many of these proteins are repeat proteins or pseudo-protein repeats. So the different packings of tertiary packings are not that different. And this again also suggests that, in fact, the energy levels of the different polymorphs are not that different, and that indeed the environment and small perturbations can really steer polymorphism. Now, what we've done in the lab is we've made a database. It's called STAMP. You can find it on our website. And what it does is actually showing, all the known amyloid structures. You can find where the APRs are in all of those. You can compare polymorphs and see how similar or dissimilar they are. And you can visualize all those structural or thermodynamic parameters and compare them between polymorphs. You can visualize the thermodynamic profile. So you can go to your favorite amyloid there and see how its architecture is determined and which residues matter most. Now, for the rest of the talk, actually, I want to go to what I was alluding to in my title. And so that's a question as to whether the proteomic background of a cell can modulate amyloid aggregation. So basically, you have an amyloid protein expressed in a cell. There are many other proteins in there. Can some of those proteins inhibit amyloid assembly? Others maybe drive it. And what happens to proteins that co-aggregate with the amyloids, and what happens to their function? And so beyond that, the big question then is, does the proteomic background, in fact, against which an amyloid is growing, does it play a role in its polymorphic structure? And does it play a role in the cell selective deposition and vulnerability of amyloids? Now, to look at that, you have to ask the question as to how amyloids can interact with their environment. And of course, we all know amyloids is a self-assembly, self-interaction process. leading to the growth of the amyloid. But if you look at the structure, you have two very different ways in which amyloids can potentially interact with their environment. One is by the flanks, which as the fiber grows become, of course, the even bigger proportion of the surface of an amyloid. And then you have the tips. The flanks, of course, they are determined by steric ridges and grooves that are formed by stacked identical residues. And they are known to be able to interact with, for instance, chaperones. They are known to be the site for secondary nucleation effects. But the specificity of this here is still rather low if you compare it to the tips, the elongation tips of an amyloid. And there, of course, the structure, what is displayed to the environment is completely different. Here you see actually the whole sequence of the core that is displayed, all the residues here. And of course, these are the growing ends of the fibrils. So they really template the docking of another identical molecule on top of this one. And so the question we went about is more looking at what else can interact on this side of the amyloid. Not that what is interacting at the flanks is not interesting, but since you have a lot of sequence information here, the question we ask then is, okay, self-assembly is notoriously favored in amyloid growth. That's why they form these nice homogeneous structures. But like everything in nature, you can tolerate small changes. And so what's happening here, if you have a sequence that have more or less the same sequence or very close sequence, will it still be able to interact, and what will be the effect? And what we did, of course, is we did mostly look at the APRs here, which are a subfraction of the amyloid core. And the reason for that is, as I explained before, these are the most stable, most rigid, if you want, more structured parts. And so you would think that entropically binding here, I mean, this could form hotspots for binding with similar molecules, homologous molecules here. And so you can then imagine different type of interactions. You could say self-aggregation, self-assembly, but you can also imagine co-aggregation or capping, or cross-seeding, different kinds of mode of interactions. And so the problem I introduced you here can then sort of be simplified, if you want, to have a protein, for instance, here, A beta, which has two APRs. Can we find proteins in the proteomic backgrounds that have homologous sequences, not to the entire A beta sequence, but just to the APRs? And can those interact? And so Katrina in our lab did this. She took the first APR here, 1621 of A beta. She just took this sequence and then she ran it along the human proteome, looking for short stretches in all these proteins that are homologous with just one or two mutations. And then she printed a microarray, a peptide microarray containing hundreds of peptides that are indeed homologous to A beta. And then she incubated those microarrays with A beta 42, biotin labeled, and so we could detect on which spots A beta was sticking and which one not. So all these spots are maximum one or two, again, mutations away from the red region here. And you can see that many interact, many also don't interact. And in fact, you can then, I mean, for instance, here you can start playing with this. These are all homologous peptides to A beta, the APR1. And you can indeed print things like AD, etc. So because some of them bind, others not. Then what you can do as well is look at how these interactions, because they interact, but then how do these interactions affect the behavior of amyloid assembly? And some of those interactions we measured many of them. I'm just showing you 2 examples. Some of them can accelerate A beta aggregation, and others can slow it down. We also looked at how co-incubation of a short homologous peptide with A beta can affect its morphology, and we did that in collaboration with Wenwei Zheng from the University of Kent, and he has this nice method whereby you can reconstruct amyloid structure from AFM images, and as you can see, just co-incubating with those short peptides really changed the mesoscale structure of the amyloids. I wouldn't go as far as saying that it changes the polymorphism of A beta. Of course, we don't know that at this resolution, and many other things can happen here. But certainly, it does change, the structure of these the structure of these amyloids. And then the question we asked as well is, okay, what happens then in a cell? And what we did is we made an A beta biosensor line, which in fact does not, where A beta 42 is expressed, coupled to a fluorescent protein, and it does not aggregate in this cell if it's unperturbed. But then if you seed the cells with A beta seeds, you will see amyloid inclusions forming. And this biosensor line nicely reacts in a dose-dependent manner to seeding. And what we did next then was we first transfected, this time full-length proteins containing a short homologous sequence to A beta, and then looked what happened. And in fact, several of those could initiate A beta aggregation just by being transfected in the biosensor line. And then we looked whether the initiation of A beta by the expression of this these homologous, regional homologous proteins was determined by those homologous regions. And it was, because indeed, as you can see, here is the amount of spots per cell you find expressing one of those proteins. And here you can see when you excise only the region that is homologous to A beta, then you really reduce significantly the effect observed. Now, of course, this is all structure-based thinking, but still theoretical in the sense that you, of course, don't know whether these things would happen in disease, whether they play a role. The only thing we can say is that we looked at deep proteomic data of human A beta plaques. And what we could see is that, of course, these plaques are mainly formed by A beta, but they contain many other proteins as well in much lower amounts, but still they are there. And in fact, in the best data sets, you can find more than 1,000 proteins, different proteins in human plaques. In this case, they are micro-dissected and then analyzed by mass spec. And what you can see is that, in fact, APRs and only APRs are overrepresented in here. So proteins that have homologous segments to these 2 APRs are overrepresented in human plaques, and other proteins that have homology to other parts of A beta are not. So really, this suggests that in real life, in A beta, in Alzheimer patients, these interactions do occur. And if you do a gene ontology analysis of the proteins that are in there, it's quite interesting to see that you really see proteins coming from the site of formation of A beta inside neurons. You also see microglial proteins in there, and then you see extracellular matrix proteins that are homologous to A beta in there. So again, the real meaning of this enrichment for disease remains to be analyzed, and we are trying to do that at the moment. But it's still very intriguing to see to see that happening. Now, I don't know how much time I have left. The couple of last slides. I just want to show here a work of Nikos in which what he did was the same thing, looking at co-aggregation, but this time by structural modeling. What he did was he took all the structures available of APRs, and for all those structures modeled all the point mutants, and see again with Foldex what would happen. And so you have different scenarios. You can have a single point mutant that still can bind to a pre-existing seed, but once it's bound, it's capping and no other can add on top of it. You can have, of course, the scenario where the peptide cannot bind at all and doesn't aggregate. You can also have a cross-seeding, if you want, effect, whereby the peptide can bind to a pre-existing tau core, tau amyloid. And sorry, this is not tau only, this is modeled on many different amyloids. And so you can have the scenario where you can dock and still elongate. And then, of course, you can have a last scenario where the peptide, the point mutant, is still aggregating, but without interacting with wild type, the wild type sequence. And so if you look at that, you can see that for all these amyloids and all these point mutants thereof, 50% of the point mutants of APRs, single point mutants, do not interact anymore. So that means that just a single point mutant here abrogates the aggregation propensity and the possibility to interact, showing how, you know, sequence specific these assembly processes are. Then you can see that about 20% and more or less point mutants can form either cappers or interact heterotypically to such cores. And of course, only 6%, it's quite nice to observe as well, can still aggregate, but without interacting anymore. So, you know, 6%, let's say, form kind of a species barrier, if you want. So, looking at those 4 scenarios in silico and on all proteins is nice to give an idea of the landscape. And I don't have time, but it's quite interesting to look at which type of mutations lead to these different scenarios. But what Nikos did was he took PHF6 of tau, which is a 6 residue peptide, and he made all the point mutants of this sequence, and then measured their aggregation and compared it to the structural predictions he made before. And as you can see here in this volcano plot, you can see that actually both the inhibition or the increasing of aggregation could be nicely predicted in general. And here you can see, again, that's, of course, on the level of small peptides. So this is the wild type peptide incubated with a point mutant. And you can see that some of them inhibit nicely the aggregation of the peptide, others increase the aggregation. And you can see that the one that inhibit aggregation also increase the critical concentration of the peptide. And conversely, the one that favor aggregation will also lower the solubility of the peptide. And again, this is at the peptide level, but what Nikos did afterwards is using biosensor lines again with full-length proteins. So he expressed proteins bearing such homologous sequences to PHF6 again, and then added tau seeds. And what he could observe is that for several of them, actually, you can really see an increase in the fraction of cells that contain spots, and also an increase in the fraction of cells with spots. So again, having a background expression of a protein that contains a short homologous sequence can again influence the assembly kinetics of your amyloid quite significantly. Nico didn't do, and this is my last slide, I think. Nico didn't do the same work with Wenwei Zheng as Katerina did on A beta, but what he did was he looked at how the amyloid structure of tau PHF6 varied when incubating with those peptides, with those homologous peptides, and he did that using curcumin or PFTAA and looking at how the spectra changed upon co-incubation. And then by clustering, you can really, this is principal component analysis. You can do other types of analysis, but it really nicely shows again that the structure of the amyloid is quite disturbed, actually, perturbed by this co-incubation with homologous sequences. He did the same with FTIR, and as I showed you in biosensor lines, you really have quite significant effects. So in summary, and I will stop here, what I try to show you today is that amyloid cores are not homogeneously stable, but contain regions that are structurally frustrated, that APR still constitute the more stable regions of amyloid cores across polymorphs, suggesting a framework model for amyloid polymorphism. And I also try to show you how heterotypic amyloid interactions can modify amyloid assembly kinetics, solubility, fibril morphology, and seeding. And with that, I thank you for listening, and I'm happy to take questions.
Thank you, Frederic, so much for your really interesting talk. I learned a lot of things, really, and I will double check all the papers that you suggested and also the website. It looks me very, very interesting. So I think that there are some questions in the QA box. Yes, we have. The first question is from Ashutosh Tiwari. Very interesting work. Can you comment on the relationship between the different mutation in PHF6 leading to variation in aggregation kinetics and its association with cellular toxicity?
Yeah, well, I'm not sure I entirely understand the question. What I can, in terms of toxicity, I wouldn't say we looked at it very much. And so far, what we looked at most was how strongly the kinetics and the seeding is affected in cells. So as you've seen, you see the biosensor line. These are the classical Diamond Lab cell lines. And you can see that with some of them, aggregation is strongly increased. Now, what I can tell you is that some of the proteins we used are relevant to disease. And we are trying now to look into mouse models whether indeed toxicity and things like that are increased. But I couldn't tell you more at this stage. Is that?
Okay. I don't see any further question, but I would just one question from my side before I will leave the floor to Magda that will introduce the second speaker. My question is, you described the amyloid brain regions in a water solvent, in an aqueous environment. But what about in the presence of a membrane-like environment, which is known to seed also amyloid aggregation? Do you think that these APR regions are the same in membrane environment or they are different?
Yeah, lipid interactions will have a big effect, of course, but I think it wouldn't change the regions that are really driving the aggregation process. I think that it's really what might change. I think what might change is depending on which regions will dock to the lipids. It might affect polymorphism, for instance. It will certainly affect the rate of assembly. I mean, the structure, many things can be modulated by that, but what I don't think will change is which regions drive the assembly process itself. So what I mean is, if you would make those mutations that abolish APRs and you would test them in an aqueous solution or in the presence of lipids, I think it would in both cases be impaired.
Nice. Thank you. And now I will leave the floor to Magda.
Thank you, Danilo. Thank you, Frederic. I have a question for you, but I'll ask it after we finish, just for the sake of time. You don't mind staying for that talk?
Sure, sure.
Yeah. So it is my pleasure to introduce Dr. Wenwei Zheng. He is an assistant professor at Arizona State University. Before joining Arizona State University in 2017, Dr. Zheng obtained his PhD in chemistry at Rice University, focusing on developing machine learning methods for analyzing molecular dynamic simulations. He got into the field of intrinsically disordered proteins in 2014 as a postdoctoral researcher in Robert Bass lab at the National Institute of Health. His research mainly focuses on developing computational methods to understand the conformational preferences of intrinsically disordered proteins and their relevance to their functional or pathological behaviors. I don't want to take much more of your time. Thank you so much for joining us today. The podium is all yours.
Thank you so much, Magda, for the introduction, and thank you for everyone for coming here. Actually, I have to apologize first. My topic might not exactly fit the symposium here because I know this is an amyloid symposium. So I was thinking about why I got invited, but I think there's something that I can tell you in a little bit about our understanding of the disordered protein, but mostly in a completely different perspective, more about of the formation of membranous organelles, liquid-liquid phase separation, and also what we are currently interested in, which is the word that I highlight in the title, transient interactions. So before I start the talk, actually, I just want to quickly acknowledge the people that's involved in the talk because I always forget at the end. And my students, Samuel Ward is the one who did most of the work, and our collaborators, Hagen Hoffman's lab from Wildman and Situn Young's lab from Case Western, and also the funding and the computers. And we are also actually actively recruiting the postdoc and graduate students if you are interested. So the first thing I want to introduce, which seems to be a slide which is not necessary here, in our intrinsically disordered proteins. And what we are interested in is the disordered proteins, and they are involved in a number of biological relevant processes, and they are not rare in sequence perspective. From bioinformatics studies, we know that they are very common. And for some of the organelles, actually, they can be about 50% of the entire protein sequence. So a few of the phenomena that has been very well studied in the field, for example, the first is coupled folding upon binding. It's saying that for disordered proteins, and they might not have structures at the beginning, but when they bind to their folded partners, they start to form the structure. And then there's also the allosteric effect when we have a few partners and for those disordered proteins, and there's very nice allosteric regulation between those non-local long-range interactions. And at last, and this is the topic that has been got a lot of attention for the last couple of years, which is the formation of membranous organelles and also the liquid droplet. And usually, there are many labs actually see very nice droplets. Their main component is the disordered protein, and they look really like liquid, and they can do fusion. they seems to be the mechanism of forming the membrane organelles. Okay, so this is the only slide which might be a little bit relevant to amyloid. Okay, so when people look at the liquid droplet, a lot of the discussions happens behind that is to say that the droplet might be the precursor of doing aggregation. I don't want to give a direct comment about that, but there's a lot of publications along that line. On this slide, I just only collect a few of them. The most famous example, the first example is FUS, fused in sarcoma, on the top left side, and we see that there's all those very nice droplets from Nicholas Fauci's lab. And then on the right side, with a single patient-associated mutation and with different incubation time, the wild-type FUS actually only form the droplet, which is functional relevant, but the mutant FUS with only one single mutation of G156E, and they start to incubate and see those aggregates, see actually fiber formation. And there are all the other proteins, almost currently most of the aggregation protein, and it might be relevant to phase separation at some point, and people always observe phase separation. And there's a lot of discussions behind that about the relation between phase separation and aggregation. Either phase separation might drive or induce aggregation, or maybe it's not relevant, but it's currently in the discussion. And what actually we have been doing for the last couple of years is actually to make models, computational models, to understand, to investigate molecular mechanism of the phase separation. There are roughly 3 different types of models, and I will very quickly go through them. The first one is all atom models. All atom models, we have all the atoms inside the simulation, and we have explicit solvent there, and we can do the simulation. But what all atom model can provide, this is just a quick example in showing that this is the side chain interactions between different type of amino acids. Oh, I completely forget I should put the color bar there. What's the meaning of that? Because I peel off from a paper that we published recently. And the blue line actually is the hydrogen bond propensity, and the red line, the red bar, is the sp2 pi interactions. And I remember the black one is actually from the black one is from the cation pi interaction, and then we also have the salt bridge, which is magenta. So actually, all atom simulations can tell you exactly what's the interaction modes involved in a phase separation, and we can even make those nice pictures to see that pi stacking, how it stacks. It does not stack completely from the top to the bottom. We know that from quantum calculation, it should stack with an angle. But the problem for the all atom simulation is clear. The first is the force field might not be accurate enough to capture all those weak interactions, because they are not that strong as what's usually seen in a in a folded protein, or even in our amyloid fibers. And they are very weak interactions. They might all contribute to the phase separation. Then it has a very high requirement to the accuracy of the force field about specific interactions between a pair of amino acids. There are a lot of work in the field actually trying to optimize the force field of the disorder proteins in the last 10 years. And the second question is also obvious, which is the sampling, because disorder proteins, they are usually quite large. For doing simulations, it's almost impossible to do all atom simulation maybe in the 10s or 20s microsecond. That's the limit, unless we go to a special computational resource like Anton. And then there's a clear problem of the sampling. We might not be able to see the relevant events that we want to see. So that's why many of the times, actually, we go to coarse-grained simulations. Coarse-grained simulations, there are different level of coarse graining, and the one that we use most common is the 1 beat per residue model, because it really balances the computational efficiency and also the residue level details that we can provide. Since every amino acid is a beat there, there has been a lot of works from our lab and also from our collaborators and also from the field for the last couple of years in trying to use the coarse-grained model to understand the liquid-liquid phase separation behavior. What we have been introduced currently is the sampling is partially solved. I would say why it's partially solved. We can actually generate those nice phase diagram, like a closed loop phase diagram or hourglass phase diagram. Very quick word about phase diagram. The y-axis usually is the temperature or some other physiological relevant variables like salt or crowding agents. and the x-axis is the concentration. And then we see that for the protein, there can be two different arms along the y-axis. The first arm is the low density phase, and the second arm is the high density phase. And they can coexist at the same specific condition. For example, here is the temperature. And then for the closed loop event, below which temperature, the first critical temperature, it does not have phase separation. And above that, it has phase separation. And then above the second critical temperature, there will be one phase again. These kind of behavior has been actually observed in copolymer phase separation, but now it's in the disorder protein. The second one is usually hourglass, and the sequence actually determines what's the difference between these two diagrams and which sequence might prefer to pick which one. And usually hydrophobic amino acids rich sequence would prefer to have a closed loop phase diagram, whereas polar and charged amino acids would prefer to have an hourglass phase diagram. So this is the coarse-grained model. And of course, the problem is clear. Even though we can partially solve the sampling error, there's still a lot of problem with the force field accuracy, because now we only have a limited number of parameters for each bead amino acid. And then it's very hard to actually capture generally all the experimental behavior of, for example, phase separation. And the last type of model is polymer model. And why I like polymer model a lot is because, yes, there's no sampling. It's very fast. Usually there's only analytical equation. The idea behind that is if we have a sequence, and then we can think about if we can reduce the complexity of that sequence, assuming there's no specific interactions, all the units, they're repeated and they're the same. And then we can use homopolymer model to describe the sequence. There's a lot of homopolymer theory that we can use that can directly provide us the analytical equation to understand the conformational properties of such kind of a homopolymer. And for homopolymer model, the first thing that people usually think about is the Fleury's famous book, Polymer Chemistry, and it's Fleury's scaling law. The idea behind that is if we have a size of the chain, and there's a scaling behavior between the size of the chain and the chain length of the chain, using that equation, I is the distance between, for example, the two ends of the chain, and then n is the chain length. And that nu on the top right corner, and this is not v, this is nu, the Greek letter, that nu on the top right corner actually tell us the scaling behavior of the disordered protein. If nu is equal to 1, it's more like a linear rod. It's just saying that the size of the chain actually is linear to the size, to the number of units that I put in the chain. If nu is 1/3 on the other extreme, this is more like a sphere. So usually for the folded protein, it's very close to a 1/3 situation, or maybe a little bit around that. But for disordered protein, there are two other numbers that's very close to the disordered protein. One is nu equal 0.5. It's a random coil. It's a Gaussian chain. And the other is 0.588. It's an excluded volume chain using a self-avoiding worker model. So from the experiment, how people actually obtain nu, actually there's a paper from 2012 from Ben Schuler's lab, and they actually observed for 6 different IDPs. For each of them, they can put different thread pair labeling position at one specific IDP. They can move the label position and trying to fit an equation for each specific IDP. They find that nu is roughly from 0.45 to 0.65 for typical disordered proteins. So there's a very large range of nu for disordered protein, and they correspond to close to a random coil to an excluded volume chain. But for the simulations, this becomes more straightforward to obtain. We can quickly obtain the entire trajectory of the simulation. Then we can calculate the distance as the average, the root mean square distance between the I's and J's amino acids. And then we can use that equation to fit the nu using the sequence separation and the distance relation. And a very quick example using a very old simulation I run in 2016. This is actor at different denaturant concentration. And we see that with the distance as a function of the sequence separation, we can very nicely, even in all atom simulation, there's enough sampling to obtain that special parameter nu for the disordered protein. And in 2019, we collect all our existing all atom force of all atom simulations in the specific force field AMBER 03WS. Actually, in that force field, we collected about 50 different kinds of disorder protein for the last couple of years, and we find that the scaling exponent is roughly in that range from 0.45 to 0.65. And we also know that for disorder protein, this is also have been seen in the experiment. Disorder protein, the scaling exponent, the scaling behavior, the average is about 0.5, which is close to a random coil. So why we talk a lot about the polymer model? Because for the polymer model, sometimes it can give us very nice insights and a very simple insight about what's happening. Just use phase separation as an example. In the polymer world, there are two temperatures that people know that they correlate with each other. The first temperature is the critical temperature of the phase separation. And for example, if we just have a simple vapor-liquid coexistence, and then we have a critical temperature, upper critical solution temperature and a lower critical solution temperature, we have two for that simulation. But there's another temperature, which is the temperature at which the scaling exponent is equal to 0.5. This is what in the polymer world called theta solvent temperature. And then for two different type of simulation, one is the coexistence simulation, one is just a single chain monomer simulation, and for the collapse and extension of the protein. And we can compare the theta solvent temperature and also the critical temperature. And then we find that most of the time they correlate with each other. So if we pick a number of those proteins and their mutants and run a simulation, and they correlate very nicely. And this is a trivial result in a polymer world. For a homopolymer, we know that these two temperatures actually they correlate with each other. But for a heteropolymer, even for a heteropolymer in the coarse-grained model, these two temperatures still correlate with each other. That actually tells us a very important suggestion for this model is to say for phase separation, sometimes there's no specific interactions. Most of them, they are weak interactions. The averaging interaction strength of a polymer or of a disorder protein actually tells the behavior of the phase separation. With that in mind, we also introduced very simple model trying to find how can we predict directly from the sequence to the scaling exponent, and taking advantage of Kinshaw-Gauss lab's previous finding using the sequence charge declaration, which comes from the charge patterning. And then we introduce another parameter, which is from the hydrophobic patterning of the sequence. It's just about how you arrange the charged amino acids and how you arrange the hydrophobic amino acids. Then we generate many of the coarse-grained simulations to those random sequence of disordered sequence. We find that using a simple multilinear regression of these two parameters, charge patterning and then the hydrophobic patterning of the sequence, we can actually predict the scaling exponent very nicely. So that's saying that now we have a very nice workflow for the phase separation, for predicting the liquid-liquid phase separation and the critical temperature. If we start from a sequence, and then we can run the coarse-grained simulations using the SLAB simulation method, we can obtain a phase diagram, or we can directly save the trouble. We can just run a single chain simulation and get the scaling exponent, and we can predict using the correlation between the theta solvent temperature and the critical temperature. I'm not saying that every time the correlation works for a specific system, but usually it works fine if there's no specific interaction. And we can also use very simple models to directly jump that framework. Starting from the sequence, we can use empirical equations, for example, from SCD and SHD, and also from new, for example, there are new path, PARSC. This is a software from Lauren Ho and Stephen Witten's lab, and they can all help you directly predict the scaling exponent from amino acid sequence. Then we save the trouble of running even the simulation. That can quickly give us a framework to do the high throughput screening of the sequence, tell its critical temperature. And if we want to have finer details, we can still run the all-atom simulations and trying to find out what's the specific interaction that's involved in the phase separation. However, so my talk today is mostly not about this, but about transient interactions. We know that. So the question I want to ask is, for phase separation, there's not that much specific interaction. So that's why we can use this simple workflow, directly starting from the sequence using homopolymer model, and we can predict the scaling exponent and even the critical behavior. But what if there are specific interactions beyond a background of these non-specific weak interactions for disorder protein? And there are two examples through the collaborations that we have done for the last couple of years. We start seeing more and more important roles of the transient interactions or those specific interactions for IDPs. The first example is when we are in the collaboration with Hagen-Hoffman's lab, studying the disordered tail of E-cadherin and interacting with folded beta-catenin. So the protein, actually, the disordered tail bind to beta-catenin, and beta-catenin bind to alpha-catenin, and then at last bind to actin. And then there's the extracellular part actually providing the linkage between the two cells. So for that disordered tail of cadherin, and we find that if just looking at the crystal structure, which has been solved for quite a long time, and the crystal structure inside the same unit cell, there are two structures, the left one and the right one. And the two structures, the folded part does not change, but the disordered part, when it binds to the folded part, and if I draw the sequence plot, and the first structure from residue 56 to 112, it's structured, but the N-terminal and the C-terminal part, they do not have structure. But for the second structure, the middle part of the chain, it does have the structure, but the structured part is a little bit different from the structure one, whereas for the C-terminal tail, it still have the structure binds to the folded partner. So because of that, it motivates us, and also Hagen-Hoffman's lab actually trying to think about how can actually the disorder protein binds to its folded partner, but there's still such kind of structure flexibility inside the folding upon binding case. How does that provide high affinity? Because usually we imagine that with such kind of flexibility, there will be very weak binding, which is not true. Actually, for this protein, the binding affinity is in nanomolar regime, which is very strong. So how can that provide specificity and high affinity and flexibility? So that's why Hagen-Hoffman's lab, actually their expertise is doing FRET. FRET is they can put two dyes onto the two sides of the protein, and then the distance between the two dyes is proportional. There's a relation between the FRET efficiency, it's the signal from that experiment, and the distance between the two dyes. And using that equation, Foster equation, and they can actually change the dye position because of those two crystal structures within the same unit cell. And they try to cut the protein at different parts and cut the protein into three different segments, A, B, and C, at 44 and at 119. And then there are four different labeling positions. Because for FRET, they can only do pair labeling. So if we just do a combination problem, there are 6 pair labeling positions, namely from 1 to 44, 44 to 119, 119 to 159. And then from between A and B segment, and between B and C and the entire segment, the entire protein. With that 6 different segments, pair labeling position, they can actually obtain the FRET efficiency of a number of these situations. And then we can use a very, very simple coarse-grained model that we developed for phase separation. There are only 3 different functional forms: bounded, non-bounded from amino acid hydrophobicity, and also the Coulombic interaction using Debye-Huckel electrostatic screening. And then we can actually reproduce very nicely the experimental behavior. So very quickly, the solid line is from the experiment, and the dotted, those circles are from the coarse-grained model, and the dashed line is from the polyamphiolite theory, which is a mean field theory. So that's saying that for the 6 different pair labeling positions, and all of them, 5 of them, the salt actually induced collapse of the protein, except the A segment. The salt actually introduced an expansion for the protein. So that's why we actually trying to go back and look at the coarse-grained model and find out why specifically for that A segment, polyamphenolite theory, the mean field theory breakdown, and also the coarse-grained model can still capture that. And we find this very nice contact map. This is not exactly the contact map, but in the same behavior. And we can actually plot the scaling exponent as a function of the distance, as a function of the residue number. And then we find that for the entire chain, the scaling exponent can differ from 0.42 to 0.62. And on the top, this is at low salt concentration. For the A segment, we see very nice behavior in the A segment. On the N terminal and the C terminal part of the A segment, there will be attractive interactions that give us the behavior, which is why the salt actually introduce expansion for that protein. At high salt concentration, it looks more like a homopolymer. There's no such kind of specific patterning in the scaling exponent map. So this is actually tells the first situation is we find that for the tail of the coarse-grained model, it cannot capture the Fred efficiency. That's due to the sorting out effect, which will be resolved in the next 2 slides with a new model. And the second, which bothers us quite a lot, is the transient interactions we see in this disordered protein. Because usually for Fred efficiency, we can use a self-avoiding Walker model to quickly analyze the distance between the 2 parallel di-labeling positions. But with these transient interactions, the homopolymer model will break down. And this is the main topic I will be talking about for ongoing work. So the first is the sorting out effect. Sorting out effect is just to say that usually for salt, there can be 2 different regimes. At low salt concentration, we usually see electrostatic screening that can be captured by simply a Debye-Huckel screening exponential equation that we can use combined with the Coulomb interaction. And the second part is the sorting out behavior. And recently, we actually trying to dig from the old literature, the sorting out constant. The positive sorting out constant means that if we increase the salt concentration, the solubility of those proteins will reduce. And then a negative sorting out constant means that if we increase the salt concentration, they could become more soluble in the solution. And we dig out into the old literature and find that for about half of the 20 amino acids, we can get the sorting out constant from the old literatures. And then for the other half, 10 amino acids, we trying to find a correlation between the sorting out constant and the amino acid hydrophobicity. We see a very nice correlation. Then we can extrapolate and trying to determine the sorting out constant for all those amino acids. Then we can embed that sorting out constant into the coarse-grained model in a concentration-dependent manner, and we can reproduce that tail from the coarse-grained simulation. Now we can capture that tail. So note, so this is the x-axis, the salt concentration. I'm plotting that in a logarithm scale so that we can see the tail, that deviation. And with the old model, without the sorting out constant, we cannot actually capture that. With the new model, we can capture the tail. Now we want to quickly do a verification of that. And we're trying to find all the existing, in 2021, we find 17 proteins with salt-dependent phase separation. So for some of the protein, when people increase the salt concentration, it will phase separate, and we call them high salt phase separation. For some of the protein, if we reduce the salt, and experiment observe that they will phase separate at low salt concentration, we call them low salt phase separation behavior. And for some of them, people see dual phase behavior. That's saying that at low salt and at high salt concentrations, there's no phase separation, whereas only at medium salt concentration, there's phase separation. So we can quickly simulate the radius of gyration as a function of the salt concentration for the new model. When we have a salt-induced collapse, a smaller radius of gyration, the size of the protein means a stronger interaction, means they prefer to phase separate. And then the bar here, for every plot, this is the experimental condition for determining the salt-dependent phase behavior. We see that for the 17 case, we can capture 13 case, and for the other 4, and we cannot capture, which we still don't know why, but actually introducing that sorting out effect is important to capture, especially the high salt phase separation behavior, which cannot be captured by the original model. So this is more like a sidetrack that we have learned from the Fred experiment. Getting back to that Fred, the E-cadherin and beta-catenin complex. Now, Hagen-Hoffman's lab, they are able to add beta-catenin into the disorder protein and trying to find out for the complex what's happening. So for this complex, if we want to use a coarse-grained model to capture that, that's very straightforward. We can introduce another term, which is just a native interaction term telling the folding upon binding behavior. But clearly, there's a free parameter for that complex. There can be two different modeling strategies. The first is we can assume the complex is completely rigid. And the second is we can say it's flexible. So we can tune the interaction strength of forming that complex. We find that in order to match the Fred efficiency of that disordered tail within the complex, we have to use a very flexible model to actually match that for the Fred efficiency. If we want to quickly check what's happening inside the coarse-grained model and what's happening between the folded partner and the disordered protein, we can use a cylindrical coordinate and assuming the folded protein is in the middle, and then we have a main axis for the folded protein. We also have a rotation degree of freedom. So that's saying that the disordered protein can actually move along the folded protein, either using the y-axis, the z-axis in the cylindrical coordinate, or doing the rotation around the folded principal axis. And if we do that analysis and in the structure, we can quickly actually align all the structure so that we plot all the segment A conformation, B conformation, and segment C is the C-terminal tail. And we find that for the middle part of the protein, where both of the crystal structures find that they are very stable, and we see that the big B segment almost completely align on the folded partner, whereas for A segment, it's completely flexible, even in the bound state, and C is partially flexible. And if we check all those ionic strength dependence between these two coordinates, we also see at low ionic strength, there is a barrier. There are two peaks for the distribution. That's saying that the A segment can really flip from the top to the bottom of the folded partner. It can actually move along the folded partner even in the bound state. So it's saying that there's still strong flexibility even in the folded complex. This is quite different from the behavior that people usually think about folding upon binding. Even though part of the B segment, it folds, there's still flexibility there. It can move a little bit around the folded partner. So what's more surprising is we can actually try to check the interactions between those complex inside the complex. This is a contact map between the folded partner on the y-axis, or between the disordered part on the y-axis and the folded partner on the x-axis. And we find that except for the B segment, even for the B segment in that region, and the probability of having the contacts the largest is only about 70 to 80%. So that means for this complex, there is specificity, which comes from the B segment. There is also the high affinity, which comes from the entire chain with all these weak interactions and also with the B segment. And it still has extreme disorder in the complex. And it's more important, there's also those transient interactions even in the complex. And these transient interactions are extremely difficult to model. We know that. How do we predict that? That will be the problem that we'll be talking about. The other example that we actually find for transient interaction is in a recent collaboration with Sichuan Yang's lab from Case Western. We are actually looking at the N-terminal domain of estrogen receptor alpha. And this is a fully disordered domain. And this is the sequence. The blue actually is charged amino acids, and the red is negative or positively charged, and the red is negatively charged. And the cyan line, this is the aromatic amino acids. And the gray line, these are hydrophobic amino acids. If we only look at the sequence, and if I want to make a model for this protein, and we will see that there's a bead here, there's another bead here. They are relatively attractive due to their attractive interactions of the blue bars and the red bars. And there are a lot of aromatic amino acids in the middle. So that means this is more like a dumbbell. On the two sides, this is electrostatic driven attractive interactions. And in the middle, these are aromatic amino acids. They might give a hydrophobic collapse. So Sichuan Yang's lab actually applies 3 different experimental technology. The first is small angle X-ray scattering that roughly tells the size of the entire protein. And the size of the protein is relatively collapsed compared to the other disordered protein. And the second and the third, these are the 2 experimental methods. Hydroxyl radical protein footprinting actually tells roughly the solvent accessibility of the labeling position. And then for F19 NMR, using the chemical shift of NMR of F19 label at different deuterium solution, actually they can find the solvent accessibility of fluorine label at that specific position. So HRPF and DFCS, those 2 experimental technologies they are using, they are actually labeling different parts of the disordered protein, and they can obtain the solvent accessibility at almost the entire protein, different position of the protein. And the problem is this is a disordered protein, even with those about almost 24 different labeling positions, it only gives us limited number of restraints to determine the interactions inside the proteins. But even with those restraints, we find that a coarse-grained model with only electrostatic interactions, with only Coulomb interaction is hydrophobic interaction, it's not possible to capture all these finer details. So that's why at last we go to an all-atom model. And using an all-atom model, the current force field, if we just look at the initial ensemble generated from the force field, it looks terrible. So we have to do an ensemble fitting by reweighting the conformations in the all-atom conformation and trying to find out what's the best ensemble that we can match those experimental data. After doing an ensemble fitting procedure, we can match all these 3 experimental technology, small angle X-ray scattering, and then protein footprinting, and then F19 NMR. And for the next 2 experimental technology, we can only see the correlation because they do not really tell us the solvent accessibility. They only tell us the relative solvent accessibility of the labeling position. With those restraints, a lot of restraints put in the all-atom ensemble, we can check the contact map from these all-atom ensemble. And we find that again, we see those non-local, long-range transient interactions. And they actually match exactly with the solvent accessibility of some of those amino acids, because in order to capture the low solvent accessibility of those amino acids, they are pushing the all-atom ensemble to actually introduce those interactions between non-local amino acids with large sequence separation. And on the right side in the bottom, I picked the top 3 configurations with the highest weight, but these are just representative conformations because we know that there are more than 1,000 conformations contribute to that ensemble with the flexibility. And we see between amino acid 20 to 60 roughly, and amino acid 100 and to 130, which is colored by blue and red, they have these transient interactions inside the structure ensemble. But for many of the structure, it does not exist. So that's why at last, there's about 10 to 20% of interaction. That is sufficient to explain those experimental measurement telling the solvent accessibility. So for both of the projects, with the collaboration, we find that there's transient interactions beyond those non-specific weak interactions. The first is a disordered protein bind to a folded partner. But if we only look at the disordered protein, that segment A, and we find that those transient interactions, they are introduced by charged amino acids and highlighted in yellow and red. So on the N-terminal part, there are 3 arginine, and on the C-terminal part, there are every 3 fragments of the charge patch negatively charged, and they have attractive interactions, introduce those transient interactions. And on the right side, for the estrogen receptor case, if I highlight the part that actually have interactions, we can see limited evidence of charge-induced transient interactions, but it's more like hydrophobic induced. So that's saying that how can we capture those transient interactions using a simple model? The motivation behind that is for both all atom simulations and coarse grain simulations, if we want to explain that explain experimental technology using these models, there might be over-interpretation because of the flexible conformation, because of the ensemble properties. And we have so many degrees of freedom that cannot be determined by limited experimental restraints. So that's why we want to find if there's a as simple as possible model with limited free parameter, so that we can use that to analyze limited experimental inputs. And the first thing we want to know is we have to think about how can we have a reference system where we have transient interactions in the model so that we can have tunable transient interactions so that we can train the model. So the idea behind that is we quickly check the displot database with all those disorder proteins. And of course, we take out those repeated ones, and we only check the ones starting from amino acid sequence chain length from 30 to 400, so that homopolymer model can still apply to that. And we're trying to find the frequency of those charged patches and to see what's the most probable patch length of disorder protein. And we find that starting from 2, that's saying that there are 2 positively charged amino acids. They are set together with each other in the sequence for a disorder protein. And then 4 until 8, we find that from 2 to 6 for the disorder proteins, and it's still quite probable to have those fragments of charged patches in the disorder proteins. With those patches, there is a possibility that they will introduce transient interactions between them. But we also check the reference line to see that whether these stuff, they are designed by evolution or they are just random noise. So these 3 dashed lines, they are the one that if we assume the same sequence composition, we just randomly arrange those amino acids. And we find that in reality, IDP sequence for those charged patches, they are much more frequent than randomly align them, randomly distribute them in the sequence. So that's saying that there might be some functional relevance between those charged patches. With that in mind, we can now design a very simple model just for us to play with, a simple model which is what we call DRN peptide, where we have the N-terminal part with D signals negatively charged and with C-terminal part with positively charged beads. And each of them, there are N amino acids. And then we have in the middle, all of them, they are glycine. And we have 100 minus 2N so that we can keep the chain length to be 100 amino acids. The reason that we want to introduce that is glycine is it does not actually interact that much with the others. Whereas the 2 patch, there might be transient interactions between the N-terminal and the C-terminal part. And then we can run coarse grain simulations at 300 Kelvin. And we find that there is really a trend of transient interactions when we increase the N. When we increase the N to about 4, DR4 peptide, there is a balanced transient interactions between the balanced transient interactions and excluded volume peak, which is the typical IDP-like interactions. So with that, we can try to find a model to capture both of the 2 peaks in a P alpha distribution. And the first peak is an IDP-like peak, which is a typical peak on the right side, and we can use a self-avoiding worker without any specific interactions. And then the second peak is the transient interaction peak, motivated by a recent work from Papu's lab, and we can just use the Gaussian function to fit that peak. By combining both 2 functions together, now we have a new model. We call it Saw-Nu transient model. Actually, we have both an IDP-like peak from a self-avoiding worker and a transient interaction peak from a Gaussian distribution. And then, of course, there's a free parameter actually tuning the interaction strength between these 2. That's the weight between these 2. With these 2 models, we can actually trying to analyze what we have, trying to fit these DR series peptide. And we find that because for the Gaussian distribution, there are a lot of free parameters, including the center of the Gaussian and also the standard deviation of the Gaussian. And we find that by fitting from DR2 to DR6, we can roughly use an average parameters of the center and a standard deviation. We can roughly capture the peak position of the transient interactions. So using that, we can effectively reduce the number of parameters for this model. Now we only have two free parameters. The first is nu, which is the scaling exponent that tell us how expanded the protein is, that actually tell us the distribution of the exfoliate volume or all different type of weak interactions, IDP-like interactions. And the second is Ft, which is the fraction of the transient interactions. It tells us how important the transient interactions in the model. With that model, why we need that? With that model, we can actually analyze all these DR peptides, and we find that at different temperatures, the transient interaction will clearly reduce clearly. So at 600 Kelvin, all of them, they disappear, but at 300 Kelvin, for different DR peptide, there's still sufficient transient interaction from those charge patches. Now we can try to find out whether we can use that model to analyze existing experimental data and how many existing experimental data we need to interpret that peak. So the idea behind that is the framework is actually the TOC figure that I'm using today, even though it appears in the 27 page. So we have a cost-grain model. We have the simulation with those P of R. And we can use those simulations as a reference and generate artificial experimental signals from those simulations by quickly calculating the averaging experimental behavior using all these distances. And then we can minimize the model between the experimental signal and the model interpreted signal. By minimizing them, we can determine the fraction of transient interactions and also the polymer scaling exponent. At last, we can compare the model interpreted P of R, the distance distribution function, with the simulated P of R. And we find that the difference between the cumulative distribution function using a KS test, we can tell whether the two distribution functions, they are comparable. If they are comparable, that means the model here that we introduce here with the transient interaction can effectively analyze the fake experimental data, the artificial experimental signal, which might be applicable to analyze experimental signal. With that in mind, very quickly, there are three experimental technologies that we are thinking.
Wenwei,
Yes.
Sorry to interrupt you. Do you mind wrapping up? We are really beyond our time. Sorry about that.
Yes. So how many minutes do I have?
You were supposed to talk for half an hour. I don't know if Rams warned you about that.
Okay. So
And
let
we
me
are
very
hitting
quickly.
an hour almost, like 50 minutes.
Okay.
Apologies,
I'm really
because
sorry.
we are losing. Yeah. Thank you.
Yes, very quickly. So we test three different experimental data with the probing lens, and different experimental technology, they are sensitive to a specific distance. For example, Fred is about 5.4 nanometer, which is related to the dye. And there are two other experimental technology that we can test. So with that in mind, I will very quickly skip and directly push to the last slide, which is very interesting. So we can actually tell that with the transient interactions, we have the transient peaks. We can say that we have two different experimental technology. They have different probing lengths. And we can test whether combined two experimental technology can help us reconstruct the P of R, the distance distribution function. And we find that if we have two experimental technology, they have complementary probing lengths. They actually can help us resolve both the transient interaction peak and also the self-avoiding Walker peak like that. But there's also another minimum, which is quite interesting, is to say that both of them, if they have large probing lengths, and they can also help us resolve both the short range peak and the long range peak. That's not mostly because for our model, there are two free parameters. If we have two experimental technologies, they have very large probing lengths. They can actually effectively reweight the fraction of the transient interaction and give us that. So at last, a very, very quick summary, because I realized that, sorry for the time. So I quickly actually introduce our new model, actually trying to find the transient interactions. And there's a lot of different situations when we collaborate and we find that there's those important transient interactions. They might be specific in the disorder protein. They might not be important for phase separation, but they can be important and functional relevant for the other properties of disorder protein, especially like folding a bond binding case. And an acknowledgement and thank you so much for for the invitation, and sorry for the time. Yeah.
No, that's okay.
Yeah.
It was getting to. I mean, I kind of wanted to have some questions after that.
Yes.
But we can. I don't see any questions. It's actually your work is extremely interesting with all these simulations and prediction. And my question for you was, and it's actually quite relevant to protein aggregation, as you alluded at the beginning, that many it's pretty much condensate formation. It's kind of a precursor on the way for aggregation. So it's extremely interesting. My idea is, and I really don't have much. This is my only question, so we can wrap it up. I know that the phase separation requires nucleation, not always, but there are occasions where it requires nucleation. And do you have, can you simulate that? Can you simulate these processes? And can you even predict when it will assemble or condensate faster under one condition or the other condition? So do you have...
Yes, for phase separation, usually there's no nucleation in our. It's an equilibrium where you actually directly have the coexistence for the two phase. Yes, we can simulate that. I think the way that we simulate is we use more coexistence simulations where we have this, and that's a very quick. So we actually say that we have a slab in the middle for the simulation. And then we can actually quickly scan the simulation at different temperatures. If there's in coexistence, then you see that those peptides will fly out of the slab. And then there's the low density phase on the two sides and the high density phase in the middle. So that can quickly tell us the coexistence of the two phase. So that's what we do for the phase separation. But clearly for aggregation, it's not as simple because there are specific interactions and the energy is quite stable. And they are not fluid-like, they are solid-like. Usually they are irreversible. So this kind of simulation is not doable for aggregation. But for phase separation, this is doable because they can quickly go inside of the droplet and go out of the droplet. So that's what I meant when I was talking. The sampling problem is partially solved for phase separation community, but clearly not for aggregation community.
Yeah. Thank you so much. I think we are ready to wrap it up. So if anybody has questions for you, they know how to reach you. Thank you so much for coming. And I really do believe that your talk was really relevant to our ZOOMinar series, because we are yet to learn all these processes, how they're happening. And that's a really great way to do it.